If you're sitting the Cambridge AS and A-level biology 9700 exams, mastering the 2.1 Biuret test for proteins isn't just, a good idea. It is an absolute survival requirement. Listen closely here because this specific topic, it appears in literally every single Cambridge examination series. There is simply no escaping it. But rather than just having you memorize a textbook, we're going to look at this strictly through the eyes of the principal examiners. In this explainer, we're breaking down exactly what examiners are actually looking for, the traps that cost students marks every single year, and how you can lock down those top 10 grades. All right, let's get right into it.
First up, let's look at a typical, flawed student answer on the left. It vaguely mentions that the solution turned purple because proteins are there. Honestly, it completely lacks specific context and scientific rigor. This ordinary answer is just bleeding marks. Now, compare that to the right side. This right here is a top-band answer. It uses highly precise terminology, specifically noting the detection of peptide bonds, and it logically structures the observation to match the biochemical reality. Immediately, you can see how costly a casual approach is. A few missing words, and boom, you've dropped an entire grade boundary.
All right, let's dive into Section 1, Core Concepts, and what I call the terminology trap. So the theoretical architecture of the Biuret test doesn't just rely on knowing the steps of the experiment, it rests entirely on precise definitions. The Biuret test is a biochemical test used to detect the presence of peptide bonds in proteins. I want to highlight that. Principal examiners point out year after year that candidates consistently confuse key terminology here. If you write that the Biuret test tests for amino acids, or just vaguely say it tests for protein molecules without defining the actual mechanism of peptide bonds, you are going to be severely penalized. Getting this exact definition right and always defining your terms before you use them in extended responses is really your first major line of defense. So, to actually conquer this, we need to understand exactly how you're being tested, and the magic number here is 3. The Cambridge assessment objectives for this topic specifically target 3 distinct cognitive domains that we absolutely must master if we want to grab those top marks. These 3 domains are knowledge and understanding, handling and evaluating information, and finally, experimental skills. You see, Cambridge prioritizes analytical depth and conceptual understanding way over just superficial breadth. They don't just want you to recite a color change like a robot. They want you to evaluate alternatives, compare different approaches, and really justify your conclusions. When you integrate this test with other syllabus areas and show that connected conceptual understanding, I promise you, your marks will skyrocket. Moving right along to Section 2, the method mark strategy. This strategy is a complete insider approach to locking down process marks during the practical portions of your exam. This is where marks are awarded for the process itself, not just the final answer you get. In a moment, I'm actually going to reveal the exact examiner clue that flips an answer from a fail to a top band. But first, you really need the framework that makes that clue completely obvious. Without this framework, you'll basically be staring at exam data completely blind. The crucial point here is mastering these 6 procedural steps. Practical work in the Biuret test develops very specific skills. Step 1, select the appropriate apparatus. Step 2, follow sequential instructions perfectly. Step 3, make accurate observations. Step 4, record your data in suitable tables. Step 5, identify anomalies. Actually, pay attention to that one. It's a huge deal. And Step 6, draw evidence-based conclusions. This procedural framework builds your method marks step by step. If you skip a step or even combine them a bit sloppily, the examiner literally stops awarding marks. Now, this is the ultimate problem-solving engine used by top-scoring candidates. It goes like this: deconstruct, solve, evaluate. When you're tackling examination questions, first, deconstruct the question to identify given information and extract relevant data from tables or graphs. Second, break it into logical steps to solve it systematically, executing with clear notation. Finally, evaluate. Top-scoring candidates demonstrate evaluative thinking by checking for reasonability, questioning assumptions, and justifying every single one of their decisions with hard evidence. Which brings us to Section 3, Top Examiner Warnings. Now that we have the framework down, it's time to escalate the stakes with the red pen perspective. You need to burn these 3 fatal mistakes into your memory because these are the exact things examiners are frankly exhausted from seeing. Number 1, students constantly confuse cause and effect in the Biuret test. Number 2, students simply forget to check units and terminology. They present correct numbers, but with missing or entirely wrong units, making the data absolutely useless. And number 3,
answers that lack specific context. You cannot just give a generic textbook answer. You have to apply your conceptual understanding to the unfamiliar context provided in that specific question. All right, let's switch gears for section 4. It's time for an interactive challenge. Let's break out of passive learning mode for a second. I want you to act as the examiner right now. Apply that deconstruct step to this mock experimental data table for a Biuret test. You've got your test tubes, your solutions, and your observations right there. Apply the problem-solving engine we just talked about. I want you to fully deconstruct this stimulus material. I'm just going to pause for a moment here. Can you spot the anomaly? What critical errors did the student make that will immediately cost them marks? Try to predict the examiner's feedback in your head right now. Got it? And here is the examiner-style correction. Did you catch it? The units are completely missing in the table header for the volume of the solution. And in that final observation, the student totally confused cause and effect. They stated the color changed because they added the reagent, rather than explicitly connecting that purple coloration to the presence of peptide bonds reacting with the copper-2 ions in the reagent. This ties directly back to those top warnings we literally just covered. This is exactly how you lose marks, even when you technically perform the physical experiment perfectly. Finally, we reach section 5, the diagnostic cliffhanger. Okay, so you now know the definitions, you know the procedural steps, and you know the precise mistakes examiners hunt for, but there is still one massive, unresolved problem standing between you and those top band marks. Before we reveal that hidden mistake, a quick reminder, make sure to subscribe to our channel, Ace the Exams, at acetheexams_IGCSE to excel in your Cambridge examinations. Making this explainer series part of your core revision strategy is exactly how you stay ahead of the curve. So what exactly is that final hidden cause of lost marks? Well, it's the command words. Knowing the Biuret method flawlessly is completely useless if you misread the exam's command words. If they ask you to evaluate and you just describe, you get a big fat 0 for that section. No exceptions. Your next mandatory step is checking out our explainer on Biology 9700 command words that actually decide your mark band. It is the absolute only way to ensure your scientific knowledge truly translates into Cambridge marks. Are you ready to see exactly how examiners use these simple words to trick you? Head over to that explainer right now and let's secure your grade. I mean, you didn't do all this hard work just to get tripped up by a single word, right?